So you've already done a lab exploring or inquiring about how water moves in and out of cells. You've taken some notes on it, but now we want to do a little bit of practice. And so if you'll have out page A23, we're going to do some cell transport practice. And so I want to make sure that you're following along with me. Please make sure you have out page A23. Where the problems below, a semi-permeable membrane is separating the two solutions. Now we used a plant as a model for osmosis because we didn't want to put an animal in there. If you put a freshwater fish in salt water, it will die. The potato that you're using, people were going to eat that anyway. And so the potato isn't really going to continue to live anyway. Here, they're not even actually using cells. They're just using a membrane. And so where do you find a semi-permeable membrane? Remember, semi-permeable membranes are your cell membranes. And cell membranes are found, around the outside of all cells. And that's really important. Even plant cells that have a cell wall also have cell membranes. What is the movement of water across a membrane called? This is called osmosis. And we do want to know that osmosis is actually passive transport. Of water. And so number 3 asks, during passive transport, how do molecules move? They move in a particular pattern, and it's from a high concentration to a low concentration. And we want to add in that we also call this spreading out or diffusion. So you need to know these words and these ideas. That if we put a cell, just like we did yesterday, so draw this with me. Here is our pure water, 100% water. So we had a cup of 100% water, and we put a piece of potato in it. Was your potato 100% water? No. So there was less water in the potato compared to the concentration of water in the cup. And so because water was high, it moved from high to low, and your piece of potato in the 100% water should have gained weight. Now that piece of potato didn't grow. We don't want to use the word grow because that means to add more cells. It doesn't grow, it just gains water or swells. And so what we're going to do here is we're going to do some predictions. We're going to do some practice. For each prediction, tell which way the water will move. Well, we don't have the water numbers given to us. We have something called sugar given to us. And we want to label that that's an example of a solute. Solute. Sugar and salt are both solutes. They take up space within the water area. So this first one, sugar is 40%. Well, how much of this space would be left? That would be 60% must be water. And so that's inside of the quote-unquote cell. We're having this little bag represent a cell. Outside, there's also 40% sugar. That means that there's 60% water. We're looking for where there's more water because water will move from high to low. Well, here the water's actually equal. So we're going to write the word equilibrium. Except we have to be very careful. That doesn't mean that water isn't moving. It means that it's moving back and forth equally. Very important. Back and forth equally. Molecules are always moving, even though we can't see them. And if it's at equilibrium, it's moving back and forth equally. Let's work problem B together as well. If we look at B, it tells us that we have 90% sugar inside of this cell. And so the room that's left, how much room is left inside of the cell? 10% water inside of the cell. Outside, we have 40% water. That means that outside is 60% sugar. Now keep in mind, sugar cannot move. And so it's trapped. The sugar isn't going to be able to move because this membrane is semi-permeable. It controls what enters and exits. And so what's going to move instead is the water. So our prediction can start with water will move because the sugar can't, and we're not equal. So something's going to move, it's going to be the water. Water is small enough to fit through the pores, the little membrane proteins. And so which way will water move? That's a good question. Water always moves from high to low. So where's there more water? Outside. And it's going to move inside. So water will move into the cell, and the cell will swell. So what you have to do left is one more problem and then some questions that you're going to answer to go along with this. So make sure that you have completed your the rest of this sheet, and then you're actually going to be going back to the lab on page A26. So pause the video here, and then we're going to go back to the lab on page A26. So after you finish the transport practice, we're coming back to your lab. And you took the data yesterday, but you haven't started to answer any of these questions. But now you have the correct vocabulary. So make sure that you're using your vocabulary words. Words like concentration. That's a really important word. Words like diffusion, osmosis. Those are going to be really important words for helping you describe what happened to the potato. So first, you're going to be analyzing your results. And then you're going to evaluate. We have a patient who is told to swish with salt water, but the patient's also considering just drinking the salt water. So you want to evaluate what would be the better thing to do. Sometimes it's really helpful to actually draw a picture, just like we drew pictures up here. So if you're not sure, you could draw a picture and label it salt water and put a cell here, and then try to figure out what will happen to the cell if you swish with salt water, and then what would happen if you drank the salt water. The last thing that you're going to be doing is comparing and contrasting. A dialysis tubing bag. So the thing we need to know about dialysis tubing is it's just like what you were just practicing with that looked like a balloon. It is selectively permeable. Selectively or semi-permeable. So only certain things can move across this bag. It's filled with a high concentration of starch and some water. The bag is placed into water and iodine is added. Our initial observations, I'm going to go ahead and give you these. Our initial observation is that the iodine in the beaker, is in the beaker only. If we look, this bag does not have any iodine. It just has starch and water. So our initial observations is that iodine is in the beaker only and starch is in the bag only. And one quick thing to remember about iodine is that iodine starts as kind of a yellow-orange. If it runs into starch, it turns a certain color. But right now, to begin with, it would be yellow-orange. Our final observation is actually going to be that the bag turns blue-black, and the bag only. into the beaker. So again, we want to use our science vocabulary. Now remember, we also have water, and water might be moving in and out. So you need to explain whether the iodine moved, whether the starch moved, and whether the water moved. But we don't just want to say move, because we have these important vocabulary words like diffusion, osmosis, and concentration. So what you're going to be doing is you're going to be finishing up the lab, and then at the end of today you'll take a quick check your knowledge quiz so that you can make sure that you understand transport.